This lecture is on Chapter 7, the nervous system. Section 1 describes the neurons and supporting cells. The central nervous system and the peripheral nervous system are the two major divisions of the nervous system. The central nervous system consists of the brain and spinal cord. The peripheral nervous system consists of the cranial and spinal nerves. The tissues of the nervous system are composed of two types of cells. There are neurons that conduct impulses but generally cannot divide, and there are neuroglia that support the neurons and cannot conduct impulses, but they can divide. I like to think that everything is connected, even though we have two divisions. It's kind of like the axial and appendicular divisions of the body. The axial would be kind of the central nervous system in a way, because that would be the head, neck, and trunk. And then the appendicular would be the peripheral, because that extends out into your appendages, your upper and lower limbs. Of course, with the peripheral, you also have cranial and spinal nerves, but the point I'm trying to make is that it extends off of that axial, and which is what our appendages do. It extends off of the axial portion. But remember, our appendages are still attached to our body, so the central and nervous and peripheral nervous systems are still attached to each other. It's still all one unit. And here in table 7.1 is going to be terminology pertaining to the nervous system. I like the way this book does this for you all. It simplifies very important vocabulary. So make sure to go in and review this, because vocabulary is essential in learning anatomy and physiology. Let's talk about neurons. The neurons are the structural and functional units of the nervous system. Their general functions include responding to chemical and physical stimuli, conducting electrical impulses, and releasing chemical regulators. They also enable perception of sensory stimuli, learning, memory, and control of muscles and glands. Most cannot divide, however, they can, in some cases, repair themselves. With the general structure of neurons, neurons vary in size and shape, but they all have a cell body that contains the nucleus, niçal bodies, and other organelles. The clusters in the groups called nuclei in the central nervous system and ganglia in the peripheral nervous system. So if you hear nuclei, when we talk about the basal nuclei, these are clusters of cell bodies in the central nervous system. And then if you learn about dorsal root ganglia for sensory neurons, that's going to be in the peripheral nervous system. The other things that the neurons have are dendrites and axons. So dendrites receive impulses and conducts a graded impulse towards the cell body, and the axon conducts action potentials away from the cell body. So this chapter is going to spend a lot of time talking about these types of impulses. This is figure 7.2 and then 7.1. 7.2 just kind of is the larger version of it. So let's look at 7.1. This shows a motor neuron, which is A up at the top here, and then a sensory neuron here at the bottom. So the motor neuron is going to be called a multipolar neuron. It's a very large neuron, and this one is going to send information to the somatic nervous system, so skeletal muscles. So these are large axons that conduct impulses very quickly down their axons, and they release acetylcholine at the terminal ends and excite the skeletal muscle fibers. Whereas the sensory neurons here in B, you can see that their dendrites are going to be on the receiving end of a sensation A lot of times these are going to be general sensors of like touch and temperature, pressure, pain, etc. And they will send information towards the cell body, which will be in a ganglia of the peripheral nervous system. And then the axon is going to synapse in the spinal cord. So we can actually kind of see a little bit of a loop here already. The cell body of the somatic motor neuron is going to be in the spinal cord, and it sends the information to the skeletal muscles, whereas the dendrites of the sensory neurons are going to be out in like the skin and things like that, and then they're going to send information towards the spinal cord. So we've got from receptor to spinal cord, and then we have from spinal cord to effector. That's kind of the nervous system loop, and then in between these will be little interneurons that go up and down the spinal cord to and from the brain. Oh, I'm going ahead of myself. All right, so let's talk about axons. Axons are going to vary in length from a few millimeters to a meter. Kind of what I just showed you in the motor neuron, super, super long. And even the sensory neuron, if it's coming from the skin on the bottom of your toe, that's a long axon there. They're connected to the cell body by the axon hillock, where action potentials are generated at what's called the initial segment of the axon. Axons can form many branches called axon collaterals. We saw that here in this figure 7.1a. And that allows them to connect to other neurons and effectors, etc. And they are covered in myelin with open spots called nodes of Ranvier. With axonal transport, this is an active process needed to move organelles and proteins from the cell body to axon terminals. The fast component moves membranous vesicles, slow components move microfilaments, microtubules, and proteins. So even though the axons are very long, they still need to undergo metabolism, and obviously we need to bring neurotransmitters, etc., down to the axon terminals. And so there will be motor proteins that will carry these vesicles quickly to those terminals. Anterograde transport goes from the cell body to dendrites and the axon. It uses the kinesin molecular motors. As I was saying, they literally carry neurotransmitters and ions. I highly recommend you looking up kinesin molecular motors. They walk. They literally walk. They use ATP to power their little feet, and they are carrying these vesicles. It's the thing that won me over biology. That right there. That was it. I was smitten. So that's anterograde transport from the cell body to dendrites and axon. Retrograde is going to be the opposite way, from the dendrites and axon to the cell body. So if it's anterograde, we're going away from the cell body. If it's retrograde, we're coming towards the cell body. So this uses dynein molecular motors with the activator called dynactin. And this carries lysosomes, autophagosomes, endosomes, and other molecules. It also can carry certain viruses and the tetanus toxin. So retrograde transport can lead to diseases if bacteria have learned to use it to their advantage. This is now figure 7.2. So this is that enlarged structure of the motor neuron that we saw up here in 7.1a. And you can see the axon hillock and the initial segment of the axon. So keep that in mind because we're going to talk about that when the action potential is generated. Then this is the axon surrounded by the myelin sheath. These are going to be neuroglial cells that make that. And they are going to insulate the axon except here at the nodes of Ranvier. And these are important because this is going to be where action potentials continue to be propagated all the way down the axon. And this is what speeds up the electrical impulses as they're conducted towards the spinal cord to the tip of your toes. Okay, so let's look at the functional classifications of neurons. they are based on the impulses, directions. And so already I mentioned that like sensory neurons are going from the receptors towards the central nervous system, the brain and the spinal cord. So we call this afferent, and I think of afferent approaches, afferent approaches. So sensory neurons conduct impulses from sensory receptors to or towards the central nervous system. So they're afferent, they're approaching. Motor neurons conduct impulses from the central nervous system to the target organs called effectors. And these are going to be muscles or glands, and so we call this efferent. And I think of E for exit. So this is going to exit the brain and the spinal cord. So sensations approach the brain and the spinal cord, and then motor output exits the brain and the spinal cord. Then we have the association or interneurons. These are located completely within the central nervous system, and they integrate functions of the nervous system. As I explained earlier in that loop, all the things that are in between the sensory neurons and the motor neurons are called interneurons, and they're connecting everything together. Because remember, the central nervous system and the peripheral nervous system are connected. Even though it's two names and they consider it two divisions, it's all very much connected. All right, let's look at motor neurons. Motor neurons have two major types, somatic and autonomic. The somatic motor neurons are responsible for reflexes and voluntary control of skeletal muscles. Somatic is perceived consciously. You're aware of it. So anytime you hear the term somatic, that's going to be something you're consciously aware of. And the autonomic is going to be the opposite of that. You're not consciously aware of it unless, you have pain in an organ or something like that. So autonomic motor neurons are going to innervate involuntary target effectors such as smooth muscle, cardiac muscle, and your glands. So you're generally not aware of those. Like maybe if you have to pee, you're aware of your bladder, but other than that, you're not aware of it. So with the autonomic motor neurons, there's two subdivisions there. You've heard of these before, the sympathetic and parasympathetic. Sympathetic is your emergency situations, also known as fight or flight, and the parasympathetic is your normal functions for resting and digesting. This is figure 7.3, showing the relationship between the central nervous system and the peripheral nervous system. It says sensory and motor neurons of the peripheral nervous system carry information into and out of, respectively, the central nervous system, which is the brain and the spinal cord. So again, we're seeing a sensory neuron coming from the receptors, and it's afferent, it's approaching the central nervous system, so it's bringing sensory information towards this brain and spinal cord. Here's one of those interneurons that's going to link the sensations up towards the brain for processing, and then you'll have interneurons that are going to bring that information back down from the brain, and it's going to connect either with a somatic motor neuron if it's going to a skeletal muscle, or autonomic motor neurons if it's going to either smooth muscle, cardiac muscle, or glands. Notice that there's going to be some ganglion here in the autonomic motor neurons. That's going to be important when you get to the chapter on the autonomic nervous system. They're going to talk a lot about that. They are going to be found in both the sympathetic and parasympathetic nervous system subdivisions of the autonomic nervous system. Structural classification of neurons are going to be based on the number of processes that extend from the cell body. We've got pseudo-unipolar, bipolar, and multipolar. Pseudo-unipolar has a single short process that branches like a T to form two longer processes. You saw that in the sensory neurons right here. I would call that unipolar in books past, but I like the way this book calls it like a pseudo-unipolar. There's only one process coming off the cell body. That's what that means. Then we have bipolar, so that would have two processes coming off of the cell body. We'll meet those in the sensory chapter. There's going to be one on either side of the cell body, and this will be found in the retina of the eye. They're very important in connecting the rods and cones and to the neurons that join the optic nerve. And then there's multipolar neurons. These have several dendrites and one axon. They are the most common. Like 99% of neurons are multipolar neurons because not only are they motor neurons, which are going to be, the somatic motor neurons and the autonomic motor neurons. But as far as I understand, most of your interneurons that connect everything together in the brain and spinal cord are multipolar. So here's the pseudo-unipolar. This is figure 7.4, and we see that the pseudo-unipolar neurons are sensory, and they have one process that splits. And so these are the dendritic branches. I found those because the arrow is showing that it's going this way. So it always starts at like the dendrite, and then it goes towards the axon terminal. Here's bipolar. So again, this is the dendrite end because it's going towards the cell body and then the axon terminal. And then here is the dendrites because, again, we're going towards the cell body. And then in a multipolar, we've got multiple dendrites coming in. And this is really important because these motor neurons are receiving input from lots of other neurons that are telling it whether to be excited or inhibited or all kind of things that we're going to learn about very shortly. And then if the action potential is going to be propagated here at the initial segment, the impulse is going to go down towards the axon terminal, down that one long axon. So pseudo-unipolar is sensory. Bipolar is going to be in the retina of the eye, and it's going to link like photoreceptors to the optic nerve. And then the multipolar are going to be motor neurons for the somatic and autonomic nervous systems, and also most of the interneurons that are going to be found solely within the brain and spinal cord. So as far as classification of nerves go, nerves are bundles of axons located outside of the central nervous system. Most are composed of both sensory and motor neurons and are thus called mixed nerves. So this is when they're spinal nerves, because as you can see, some of the cranial nerves have sensory fibers only or motor fibers only. You've got some cranial nerves like the oculomotor nerve that just moves the eyeball. It doesn't do any sensations, it's just movement. And then with the sensory, like the optic nerve, that's just going to bring in sensory information from the retina of the eye. So the cranial nerves can be either sensory or motor or mixed, whereas spinal nerves are going to be all mixed. And so a bundle of axons in the central nervous system is not called a nerve, but it's cool because it looks like a nerve because these bundles are going to have a lot of axons in them, and so you can see them. They're visible, but they're not called nerves. They're called a tract. And this is because nerves are going to extend outward away from the brain and the spinal cord, and so they have, special arrangement, kind of like a muscle would, where they have like a sheath on the outside, and they have specific bundles on the inside, etc. Whereas once they come into the brain and the spinal cord, they are actually going to be like integrated into the spinal tissue and the brain tissue, but they're still traveling in very organized bundles, which is important because, you want your highway systems to be organized or, all kind of mess is going to occur. So we call those tracts. All right, and then once more, a bundle of cell bodies in the peripheral nervous system is a ganglion, and you already saw a couple of examples of where that would occur. Like the sensory neurons, their cell bodies will be located outside of the central nervous system in what are called like dorsal root ganglia. And then a bundle of cell bodies in the central nervous system is the nucleus. And I mentioned that we're going to learn about basal nuclei when you get into the central nervous system chapter. Now let's look at neuroglia. These are cells that are non-conducting, but they support the neurons. And there are two types found in the peripheral nervous system. There's Schwann cells, which are going to make the myelin sheath that are around the peripheral axons of both the motor neurons and the sensory neurons. And then satellite cells are going to be found in the cell bodies within the ganglia of the peripheral nervous system. And this is going to be like the dorsal root ganglia where the sensory neurons cell bodies are. And then remember in the autonomic motor nervous system with the sympathetic and parasympathetic, I had mentioned that there's going to be ganglia there. So that's where we'll find satellite cells. So they're support cells that help to get nutrients, etc., to those cell bodies. And then there's 4 types of neuroglia found in the central nervous system. These are oligodendrocytes, microglia, astrocytes, and ependymal cells. Oligodendrocytes mean they have like a few branches. Oligos a few and dendros branches. So these are cells that branch several times, and they're going to form myelin sheaths around the axons of central nervous system neurons. And they can actually connect to quite a few central nervous system neurons. And from what I understand, multiple sclerosis is a disease of the oligodendrocytes. The microglia, these are going to be very tiny micro. So they're tiny neuroglial cells, and they're going to act like the immune cells because there's that blood-brain barrier that's going to bar the white blood cells and things like that from coming into the brain and spinal cord. So the microglia are going to take the place of that, and they're going to migrate around the central nervous system tissue and phagocytize foreign and degenerated material. Then we have astrocytes. These look like stars, hence astro, and they regulate the external environment of the neurons. They're going to be akin to the satellite cells of the peripheral nervous system. And then ependymal cells are like a epithelial cell almost, and they line the ventricles and the central canal of the spinal cord, and they secrete cerebrospinal fluid. They also help to circulate it because they've got like cilia on them that are going to be able to move and circulate the cerebrospinal fluid. All right, so here's a good picture. This is figure 7.5. And we see that the myelin sheaths around the axons are formed in the central nervous system by oligodendrocytes. Astrocytes have extensions that surround both blood capillaries and neurons, which helps them to regulate the chemical environment and nourish the neurons. Microglia are phagocytic, and the ependymal cells line the brain ventricles and the central canal of the spinal cord. So here we see the astrocyte looking like a star over here, connecting blood vessels to the neurons, because again, that blood-brain barrier is important. These guys are going to regulate the materials that can come out of the blood, such as, potassium and glucose and things like that, and oxygen. And then these are the oligodendrocytes. See how they branch a few times and they can actually connect to adjacent neurons, and they form the myelin sheath of the central nervous system. These are your microglia, which will be very tiny cells, and they're going to move around and phagocytize dead and degenerate material, acting kind of like your immune cells of the brain and spinal cord. And then these are the cool little ependymal cells. So this is their little roots that are going to be in the brain and spinal tissue. And then here's their cilia as it's moving and secreting the cerebrospinal fluid through the ventricles in the central canal. All right, looks like we're going to look at each one now in more detail. So microglia are going to be embryonically related to macrophages. And so, basically they have a small cell body. It says infection or trauma can lead to microglial activation, in which they become phagocytotic mobile cells. So just a review of what I taught you. There's a lot here. Let's see what else. They can divide by cell division. They can kill endogenous pathogens, remove damaged dendrites, and they may help shape neural circuits by pruning axons to eliminate inappropriate synaptic connections, help you break your bad habits. And then here's table 7.2, which is going to summarize all 6. The first 2 are going to be peripheral nervous system, and then the next 4 are going to be central nervous system. So always refer back to those tables. It's a lovely function. Let's revisit the myelin sheath now. This is the peripheral nervous system, so this is the ones that are going to be by the Schwann cells. That's named for, I believe, Theodore Schwann. He's the one, I guess, who discovered them, but they also are called neurolemma sites because they make neurolemma, which is going to be the main component of the sheath. So that's what A is telling you, all their different names. So let's go to B here, where it says these cells wrap around the axon to form the myelin sheath, wraps of the cell membrane of the Schwann cell in the peripheral nervous system, and the gaps between the Schwann cells are called nodes of Ranvier, are left open because that's where the action potentials are going to be proliferated. It looks like a tongue here that's kind of cute. And this is figure 7.6. And it's going to be this flattened, cool cell. I just call them neurolemmacytes now. That's what a lot of books refer to them as solely. Your book is calling them swan cells, which is how I first learned them. But it seems like it's moved away from that nomenclature. But nonetheless, the neurolemmacyte is going to wrap around kind of like a Swiss roll, and it's going to form that neurolemma, which is going to be kind of a fatty material, which makes sense because fat is insulating, and so it wraps around the axon. And this stays alive. Look, the nucleus is alive there, and it's going to have an important role in regenerating any type of damaged axon. Small axons here, it says, that are 2 micrometers in diameter are usually unmyelinated. However, even unmyelinated axons in the peripheral nervous system have a neurolemma, but they lack the multiple wrappings of the swan cell plasma membrane. And so myelinated axons, as mentioned, conduct impulses more rapidly. Here's a cool image, micrograph of a unmyelinated and myelinated axon together, figure 7.7. So it looks like the myelinated axon, yeah, you can see that myelin sheath, like layers there. And the unmyelinated axon is shown at the lower portion of the image. So for the central nervous system, the myelin sheath is going to be produced by oligodendrocytes. One oligodendrocyte can send extensions to several axons, and each wraps around a section of the axon. It produces the myelin sheath, but not a neurolemma, so there won't be any cytoplasm or organelles. And this is very interesting to note because remember how I said that the neurolemmacyte or swan cell can regenerate axons if there's been an injury? Well, in the case of the oligodendrocytes, they cannot. They do not regenerate. So that's why multiple sclerosis can be, such a degenerative disease, because as these oligodendrocytes are affected and die, they cannot regenerate the axon nor themselves. When we see myelin, this is going to give the axons a white color, and so they call that white matter in the central nervous system, and that's going to be the tracts. And then gray matter is going to be the cell bodies and dendrites that lack myelin sheaths. Remember I said the tracts are visible? This is one of the reasons why. This is figure 7.8, which is showing the formation of myelin sheaths in the central nervous system by an oligodendrocyte. Neat little buggers. All right. Here is the demyelinated diseases, like multiple sclerosis. So these are diseases which attack the myelin sheath. So you have Guillain-Barré syndrome. This is the T cells of the immune system attack the myelin sheaths of the peripheral nervous system. This produces rapid onset of symptoms that include muscle weakness. And then we have multiple sclerosis, which is produced by an autoimmune attack by T lymphocytes, causing lymphocytes and monocyte-derived macrophages to enter the brain and target the myelin sheaths. And so this causes demyelination. Autoimmune diseases are crazy. You don't want your awesome immune system to attack your own cells. That's bad news. Here's the regeneration, as I had mentioned, with the peripheral nervous system. So when an axon, if, we don't want when, if an axon in the peripheral nervous system is cut, the severed part degenerates and a regeneration tube is formed by the neurolemmacyte swan cells. And so growth factors are released that stimulate growth of the axon sprouts within a tube. So they kind of just like grow like little fingers towards the severed end. And the new axon eventually connects to the undamaged axon or the effector. And the swan cells in the peripheral nervous system also produce proteins that inhibit axon regeneration, but they are removed by phagocytosis during that process, so that we do have axon regeneration to occur. And there are a couple of things that are going to affect the ability of regeneration. Oh, excuse me. And so the cell body has to remain alive. And I believe that, like, the closer it is to the cell body, the more chances of regeneration there is. Don't quote me on that. Moving on. All right, central nervous system axons are not as able to regenerate. Death receptors form. That's I'm a death receptor. That promotes apoptosis of oligodendrocytes. So very sad. And inhibitory proteins in the myelin sheath prevents regeneration. And then we have glial scars from astrocytes form. And I do know that, yes, astrocytes definitely form scar tissue. And so a lot of times as brain matter dies for numerous reasons, not just multiple sclerosis, but, Alzheimer's, etc., the tissues shrink and scar tissue forms. And so scar tissue is not functional tissue. So this is not functional nervous tissue that is regenerating here. So here we see figure 7.9, the process of peripheral neuron regeneration. So A, if a neuron is severed through myelinated axon, the proximal portion may survive. So that's going to be this part here, near the cell body, but the distal portion will degenerate through phagocytosis. And they call that Wallerian degeneration. The myelin sheath provides a pathway in C and D for the regeneration of an axon, C and D. And here's our little tube there, and see the axon sprout growing. So cute. And then it can go to the effector or the injured part, as it said earlier, and regeneration has occurred. Now back to the central nervous system regeneration. That image was kind of maybe not in the right place, but all right, anyway, injury in the central nervous system stimulates the growth of axon collaterals, but central axons have a much more limited ability to regenerate than peripheral axons. So when you get the axon collateral, that's like when you could have remission, like in multiple sclerosis. There are proteins called no-go produced predominantly by oligodendrocytes, inhibit axon regeneration in the central nervous system. That's funny. It's a no-go. We are not regenerating. So the take-home message is that in the peripheral nervous system, there is hope of nerve regeneration. In the central nervous system, it's a no-go. All right, we're going to skip this and go on to electrical activity. That's enough of neuroglial. I don't want you to get lost in all the details there. That's plenty. Plenty, plenty, plenty. All right, so now let's go to the electrical activity in axons. Here we go. This is the meat and potatoes of this chapter here, because this is physiology. I believe in meat and potatoes. The fixins are nice, but the meat and potatoes are the essentials. All right, so as far as the resting membrane potential goes, neurons have a resting potential of negative 70 millivolts. And this is established by large negative molecules inside the cell, the sodium-potassium pumps, and the limited permeability of the membrane to positively charged inorganic ions. At rest, there is a high concentration of potassium inside the cell and a high concentration of sodium outside the cell. This is imperative. With the processes of diffusion, if the potassium and the sodium were allowed to just freely diffuse as their electrochemical gradients and Their diffusion gradients, concentration gradients allowed, they would equal out. And if they equal out, then we do not have a resting membrane potential. We do not have a negative 70 millivolts. And very important things cannot happen as a result. Here's what can happen. We can alter the membrane potential. So neurons and muscle cells can change their membrane potential because of this unequal distribution of ions. And this is called excitability or irritability. And it's caused by the changes in the permeability to certain ions. And ions will follow their electrochemical gradient, which is a combination of concentration gradient and an attraction to opposite charges. And we want to keep that from becoming equal. We've got to do some very special things like the sodium-potassium pump and the percentage of leakage channels that are in the membrane to make sure that they don't become equal. We've got to keep our membrane potential at negative 70, or we can either, make it more excitable by going towards what we call a threshold, or we can make it less excitable by going away from threshold. So stay tuned to that. But this electrochemical gradient and this altered permeability and the sodium potassium pump are imperative players in this game. So make sure you're kind of paying attention to them. Sodium potassium pump and different numbers of leakage channels, like different numbers of sodium channels, potassium channels, are going to be imperative. And so the flow of ions are called ion currents, and they occur in limited areas where these ion channels are located. So at rest, a neuron is considered polarized when the inside is more negative than the outside. There's that negative 70 millivolts. And again, we can go towards the threshold and become more excitable, or we can go away from the threshold. So when the membrane potential inside the cell increases, thus becoming more positive, moving towards the threshold, this is called depolarization. And so if we move towards the threshold, we are going to end up at a positive 30 millivolts. And so we're depolarized because the polarization comes from the difference in charges across the membrane. So if we're more negative on the inside, like at rest, at negative 70 millivolts, the outside is going to be relatively positive. That's polar. We've got two opposite ends. So once we change that, like I said, with the different amounts of leakage channels, there's going to be gates that we're going to learn about, like voltage gates and chemical gates that are going to disrupt this flow of ions. All kind of cool things are going to happen that will depolarize the cell and make it all the way up to positive millivolts. And this is what is going to give us that magic of the electrical current that's going to travel down the axon of a neuron. And then, when you get to the muscle fibers, that's what is going to then cause the muscle fibers to contract. But then we've got to return. We have to return to our resting potential. And so we have to get those sodiums back outside and those potassiums back inside. We need to reestablish that resting membrane potential and repolarize our membrane so that it's once again negative on the inside and positive on the outside. So I know I was excited and blurping about all that. So let's go back and take a look once more to review. So the meat and potatoes. Neurons have a resting potential of negative 70 millivolts. This is established by large negative molecules inside the cell, the sodium potassium pumps, and limited permeability of the membrane to positively charged inorganic ions. So number 2 states at rest, there's a high concentration of potassium inside the cell and a high concentration of sodium outside the cell. Cells can alter that membrane potential, and this is called excitability or irritability. And this is caused by changes in the permeability to those certain ions, particularly like potassium and sodium. Ions will follow their electrochemical gradient, which is a combination of their concentration gradient and their attraction to the opposite charges. And then the flow of ions are called ion currents, which occur in limited areas where ion channels are located. So at rest, a neuron is considered polarized when the inside is more negative than the outside. When the membrane potential inside the cell increases, it becomes more positive. This is called depolarization. Depolarization occurs when positive ions enter the cell, usually that's going to be sodium, and a return to resting potential is called repolarization. When the membrane potential inside the cell decreases, it becomes more negative, and this is called hyperpolarization. Hyperpolarization occurs when positive ions leave the cell, usually potassium, or negative ions such as chloride enter the cell. Depolarization of the cell is excitatory, that's taking us towards threshold. Hyperpolarization is inhibitory, it's taking us away from threshold. Changes in the membrane potential can be recorded with an oscilloscope by recording the voltage inside and outside the cell. So we are electricity, and we can produce our electricity through these ion gradients and the ion exchanges. All right, so changes in membrane potential are controlled by changes in the flow of ions through those channels. So potassium has two types of channels. They have leakage. Your book calls them not gated. So leakage channels, they're always open. And then there's voltage gated. So voltage gated and leakage are going to be the two types that potassium has. So with the leakage, as mentioned, they're always open. With the voltage gated, they open when a particular membrane potential is reached. So they have a certain voltage that will open them, and then they are closed at resting potential. Sodium has only voltage gated channels that are closed at rest though fast flickering occurs. The membrane is thus less permeable to sodium at rest. All right, let's talk about voltage gated sodium channels. So these channels open if the membrane potential depolarizes to negative 55 millivolts. This is the threshold that I've been talking about. So as we depolarize, we go towards that threshold. If we hit that threshold, boom, magic happens and sodium rushes in due to this electrochemical gradient. The membrane potential climbs towards the sodium equilibrium potential, and it gets up to positive 30 millivolts. And positive 30 millivolts is where basically all action potentials depolarize to, and this is because once we get to positive 30 millivolts, this then deactivates the voltage-gated sodium channels and they close. Sorry, gotta drink my coffee so I can keep going here. And I'm in the South, and so it's false fall. While I'm recording this, it's actually the equinox today, so a lot of us have like allergies and whatnot's going on, so apologize for my voice. All right, figure 7.12, voltage-gated ion channel. This is a model of a voltage-gated ion channel. The channel is closed at the resting membrane potential but opens in response to the threshold. This permits the diffusion of ions required for action potential, primarily sodium. And after a brief period of time, the channel is inactivated, in this case by the ball and chain portion of the polypeptide chain. So this is inactivated, closed. And then this is it closed at resting membrane potential. And remember, it says it can do this like flickering. It can open real quick and let like sodium in just a little bit. And that is fine. That still is not enough to disrupt our gradient. But when the action potential comes in and that threshold is reached, wham, we are open, and sodium is flooding in, and it's going to reach positive 30. Remember, that's kind of like the end game there. And once it does, boom, we're going to deactivate it. No more can come in. And so then we're going to repolarize and get back down to resting membrane potential. All right, voltage-gated potassium channels are going to open at positive 30 millivolts. So that makes sense, because if the sodium channels are closing and we're not going to have any more sodium rushing in order to reestablish that resting membrane potential, we got to get some positive ions out of there. So we're going to get those potassiums out. So potassium rushes out of the cell following the electrochemical gradient, and this makes that cell repolarize back towards the potassium equilibrium potential. We're actually going to overshoot that a little bit and become hyperpolarized, and that's going to be kind of what signals those voltage-gated potassium channels to then close And sodium potassium pump and the leakage channels are going to reestablish that resting membrane potential. Let me just get a good cough in there. Again, sorry. Doing my best. All right, so at threshold, membrane potential, which is negative 55 millivolts, voltage-gated sodium channels open, and sodium rushes in. As the cell depolarizes, more sodium channels are open, and the cell becomes more and more permeable to sodium. This is a positive feedback loop, and it causes a rapid reversal of the membrane potential. It goes all the way up to positive 30 millivolts, and we call that depolarization. At 30 millivolts, the sodium channels will close and the potassium channels will open. This results in repolarization of the membrane potential, and this is a negative feedback loop because it's bringing us back to normal. So remember, positive feedback loop is going to stimulate the change in the variable, and the negative feedback loop is going to then be the opposite. It's going to resist the change in the variable, and it's going to bring us back to our set point, our normal C, which is going to be negative 70 millivolt resting potential. Looks like we are at figure 7.13 here. So this is showing the depolarization of an axon affecting sodium and potassium diffusion in sequence. So in number 1, I don't know where number 1 is. Let's see. Sodium gates are open. Here we go. That's what I thought, the green. And the sodium diffuses into the cell. After a brief period, potassium gates open. Boom. Number 2 there. And potassium diffuses outward, out of the cell. So the inward diffusion of the sodium causes fuller depolarization. So boom, there's one again. Yay, there's one. And so here we're getting all the way up to positive 30 millivolts. And then once we get to positive 30 millivolts, that's going to close the sodium channels and it's going to open the voltage-gated potassium channels. Then we're going to get the potassium diffusing out, and that's going to repolarize us back towards the resting membrane potential. However, we're going to hyperpolarize. Where's my cursor? There it is. And that is going to then close those potassium channels, and the sodium potassium pump and the leakage channels are going to bring us back to resting membrane potential. And it does mention the positive and negative feedback stuff. So over here in number 1, the sodium rushing in, getting us further away from our set point, which would be like our resting membrane potential. That's going to be positive feedback. And then as the sodium channels close and potassium channels open, and we go towards our resting membrane potential, that's returning us back to our resting state and is thus a negative feedback. That's cool. I like the way the book relates this to positive and negative feedback. I've not seen that in the previous anatomy and physiology books that I have read. I've read a lot. This is figure 7.14. The membrane potential changes and ion movements during the action potential. Oh yeah, this is cool. All right, so the top graph depicts an action potential, the blue line. The bottom graph, red lines, are going to depict the net diffusion of sodium and potassium during the action potential. And the x-axis is the same in both graphs. It's for time, so that the depolarization, repolarization, and after hyperpolarization in the top graph can be correlated with the events in the sodium and potassium channels and their effects on ion movements in the bottom graph. All right, so here it's saying the inward movement of sodium drives the membrane potential towards the positive 30 millivolts during the depolarization or rising phase of the action potential. And again, we know that's the positive feedback. So sodium channels are open. And then once we reach positive feedback, hopefully you can say it with me now, the voltage-gated sodium channels close, and we're not going to get any more inward flux of the sodium. And so now at positive 30 millivolts, the potassium channels are going to open, and we're going to get a great outward flow of potassium, which drives the membrane potential back towards potassium equilibrium during the repolarization or the falling phase of the action potential. And we're going to go back to our resting memory potential. We're actually going to hyperpolarize a little bit, and that's going to close the sodium or the potassium channels, and then sodium potassium pump and leakage channels are going to bring us back to that negative 70 millivolts. So here's the literature about the after hyperpolarization. So repolarization actually overshoots the resting membrane potential, and it gets down to about negative 85 millivolts. So this does not reach potassium equilibrium potential because voltage-gated potassium channels are inactivated as the membrane potential falls. And then the sodium potassium pumps quickly reestablish the membrane potential and return sodium and potassium to their correct locations. And don't forget those leakage channels. Some books really focus on those, and some books kind of just whoop, but they are super important. Remember, there's more potassium leakage channels, and with the sodium, there's actually no sodium leakage channels. That's just new to me. It's the voltage-gated channels that just flicker and let a little bit of sodium in at a time. The all or none law. Once a threshold has been reached, the action potential will happen. And the size of the stimulus will not affect the size of the action potential. It always reaches positive 30 millivolts because that's when the sodium channels close and the potassium channels open. So the size of the stimulus will also not affect action potential duration. So this is going to be important when you get to the muscle chapter. You're going to talk about recruitment of motor units. And you're also going to talk about summation and tetanus and things like that. And that is going to go right back to this all or none law. So stay tuned. All right, so this is figure 7.15, the all or none law of action potentials. So a single quick shock, which is the red arrows, delivered to an axon can serve as a depolarizing stimulus. If the stimulus is below threshold, no action potential is produced by the axon. Once the stimulus has reached threshold, a full action potential is produced. Any greater stimulus does not produce greater action potentials. Thus, action potentials are not graded. They are all or none. So how do we code for stimulus intensity? As I said earlier, like with the muscles, we're going to recruit different motor units, and we're going to get like summation and tetanus and things like that. So, bam, here we go. I see the word recruitment right there. So that's how it's going to work here in the nervous system as well. So you get to learn about it right now. So a stronger stimulus will make action potentials occur more frequently, and so that's called frequency modulated. And a stronger stimulus may also activate more neurons in a nerve, and this is called recruitment. Same principle as in the muscles. So as you recruit more muscle fibers, you get stronger contractions and more force. And so as you recruit more and more neurons in a nerve, you're going to get a stronger nerve signal. So this is figure 7.16, the effect of stimulus strength on action potential frequency. The stimuli are the bottom diagram that are sustained for a period of time are given to an axon. In the first case, the stimulus is weaker than required to reach threshold, and no action potentials are produced. The top diagram, nothing. In the second case, a stronger stimulus is delivered, which causes the production of a few action potentials while the stimulus is sustained. In the last case, an even stronger stimulus produces a greater number of action potentials in the same time period. This demonstrates the stimulus strength is coded by the frequency rather than the amplitude of action potentials. So I always think about it like if you're not going to reach threshold, then it's silent, right? If it's kind of weak and you're going to get a few action potentials, we can kind of clap to the frequency there in the center. 1, 2, 3, 4. But then if the stimulus is really strong, we see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And so that's going to stimulate more neurons. And this is going to be important because This is going to kind of help us determine like the intensity of the stimulus that is coming in. Bright light, loud sound, a big change in temperature, pressure, light pressure versus heavy pressure. All those things are going to be the stimulus. Refractory periods. Action potentials can only increase in frequency to a certain point. There's a refractory period after an action potential when the neuron cannot become excited again. The absolute refractory period occurs during the action potential. The sodium channels are inactive, they're just not closed. The relative refractory period is when potassium channels are still open. Only a very strong stimulus can overcome this. Each action potential remains a separate all or none event. This prevents electricity from going like back towards the cell body, from what I've learned in the past. And so figure 7.17 is going to show the absolute versus the relative refractory periods. So while a segment of an axon is producing the action potential in this kind of yellow color, the membrane is absolutely or relatively resistant to further stimulation. So if it's absolute refractory, we have inactivated sodium channels and no stimulus can occur. However, during basically this hyperpolarization, we are relative refractory period. And this makes sense because we've reset like our sodium channels at this point. We're actually just kind of a little bit more negative than our resting membrane potential. So it's going to take like a stronger stimulus to get that neuron to fire because it's got a little bit further to go to reach threshold. So I hope that makes sense. That helps me out with that. During absolute refractory period, kind of like that protein, it's a no-go. We're already in our action potential stage, and there's nothing that we can do to get it to fire anymore. But during the relative refractory period, we're hyperpolarized. So if we have a strong enough stimulus, we can get that neuron to fire. But since we're hyperpolarized, it's going to have to be a pretty intense stimulus. Okay, so now we're at a cable property portion of the chapter. So this is the ability of neurons to conduct charges through their cytoplasm. Poor due to high internal resistance to the spread of charges and the leaking of charges through the membrane, neurons could not depend on cable properties to move an impulse down the length of an axon. So this is where we're going to get to that cool node of Ranvia that I was mentioning earlier. So this is the cable properties. We're going to basically just kind of peter out with this. So the cable properties of an axon are the properties that permit it to conduct potential changes over distances. If a stimulating electrode injects positive charges and produces a depolarization in blue at one point in the axon, the depolarization will quickly dissipate, peter out, if it doesn't trigger an action potential. The decreasing amplitude of the depolarization is due to the leakage of charges through the axon membrane, which is the dashed arrows. This results in a poor ability of the axon to conduct charges in potential over long distances. So bring in the swan cells, the neurolemmacytes, the oligodendrocytes, all of those things are going to insulate the axon, except that little like nodes, and that's going to help us to conduct nerve impulses very efficiently and quickly. So when an action potential occurs at a given point on a neuron membrane, voltage-gated sodium channels open as a wave down the length of the axon. The action potential at one location serves as the depolarization stimulus for the next region of an axon. It'll bring us to that threshold and phew, we keep propagating. So here it is, rolling, rolling, rolling. So the conduction of action potentials in an unmyelinated axon, we still will dissipate because it's going to take like a lot of sodium channels in order to continue to propagate that action potential. And so it's just going to be slow, and it's going to take too much energy, and we don't have time for that. So action potentials are produced down the entire length of the axon and every patch of the membrane in an unmyelinated neuron. It's going to be slow. So many action potentials are generated, each one at an individual event. It's just going to take too long. So we got to do myelin. Myelin produces insulation, improving the speed of cable properties. So these nodes of Renvia allow sodium and potassium to cross the membrane every 1 to 2 millimeters, and sodium ion channels are concentrated at the nodes, and this can leap the action potential from node to node. And so they call that saltatory conduction. It's just really more efficient. Again, that insulation property. I like to think of when you look at a cable that is connecting like your computer to the outlet, for instance, it's insulated, it's got rubber around it, and that's because if it wasn't, then either the figure 7.18 will occur, which is what's going to happen in your cable. It's just going to dissipate out, and it's not going to reach where it needs to go. If you touch it, it'll go into you and give you a shock, right? But, you know, if you expose those wires, then you're going to have impaired electrical activity because the energy is just going to dissipate out. Then let's say if you had exposed cable and you had, you know, different channels down that cable that could, you know, conduct that electricity, they would have to conduct that electricity at every single membrane channel all the way down that wire, and it would be real slow and you just would not get a good charge. So if you insulate it with the cable, boom, that electricity can zoom unimpeded and very directed towards the device that you are charging, that you have plugged in. And so that's going to be the case here with saltatory conduction and these nodes of Renvia that are going to be found in between the Schwann cells. So this is figure 7.20, the conduction of a nerve impulse in a myelinated axon. Because the myelin sheath prevents the inward sodium current, action potentials can be produced only at gaps in the myelin sheath called the nodes of Renvia. This is the leaping action potential from node to node, and it can speed it up by 30% in some of my other books that I have read. Ooh, this is fun. My dad taught this, and he always liked to talk about how fast these action potentials can go. And your book mentions it too. It says here, conduction rates in the human nervous system vary from 1 meter per second in thin, unmyelinated fibers that mediate slow visceral responses to faster than 100 meters per second, which is 225 miles per hour. So action potential conduction speed is increased by an increased diameter of the neuron. This reduces resistance to the spread of charges via cable properties and myelination because of saltatory conduction. Oh, look, there it is, even in your PowerPoint. Thin unmyelinated neuron speed is 1 meter per second, and thick myelinated neuron speed is 100 meters per second, or in parentheses in your book, 225 miles per hour. Wow. Okay, so this should be the last part just about. We got the synapse and we've got some neurotransmitters. I don't go into a whole lot of detail in the neurotransmitters. As you saw in one of your discussions, you get to talk about those. So let's mainly talk about the synapse here. All right, so introduction to the synapse. The synapse is the functional connection between a neuron and the cell it is signaling. In the central nervous system, this second cell will be another neuron. In the peripheral nervous system, the second cell will be a muscle or a gland, and it's often called a myoneural or neuromuscular junction. So if one neuron is signaling another neuron, the first is called the presynaptic neuron, and the second is called the postsynaptic neuron. So a presynaptic neuron can signal the dendrite, the cell body, or an axon of the second neuron. So if the presynaptic neuron is signaling the dendrite, we call that axodendritic. If a presynaptic neuron is signaling a cell body, it's called axosomatic. And if a presynaptic neuron is signaling an axon, it's called axo-axonic. Most synapses are axodendritic and are in one direction. Synapses can be electrical or chemical. Electrical synapses occur in smooth muscle and cardiac muscle between some neurons of the brain and between neuroglial cells. Cells are joined by gap junctions that span both membranes between cells and allow ions and molecules to pass from one cell to another. I kind of think of this with like the cardiac muscle. When we get into that chapter, those gap junctions are going to let the ions flow at regulated speeds between cells so that they can beat as a unit. And remember, the heart has like an internal conduction system that uses those gap junctions. So here's the structures of the gap junctions. We can see that they are called connexin proteins, and they form like little channels that are going to join the two cells. Ah, look, that's cardiac muscle. And chemical synapses are going to be the majority. So most synapses involve the release of a chemical called a neurotransmitter from the axon's terminal boutons, meaning little buttons or axon terminals. And so the synaptic cleft between the cells is very small, and the presynaptic and postsynaptic cells are held close together by cell adhesion molecules, abbreviated CAMs. CAMs. This is figure 7.22, and it is an electron micrograph of a chemical synapse. Here's the synaptic cleft, and this will be the terminal bouton, so the axon terminal. We've got synaptic vesicles. Remember, they were walked down there in anterograde fashion by those motor proteins. So scoot, scoot, scoot. And then we have the postsynaptic cell, and then in this case, that's going to be skeletal muscle. So this is a neuromuscular junction. Release of the neurotransmitter. Neurotransmitters are enclosed in those synaptic vesicles. So this is how they get released. It's pretty cool. So number one, when the action potential reaches the end of the axon, voltage-gated calcium ion channels open. So you've heard that calcium is important for lots of stuff. It's not just in your bones. It does, make the calcium phosphate hydroxyapatite crystals of your bones. But you have to have serum calcium, blood calcium, that is going to be maintained at a set point. So the reason you have to have blood calcium is because it is imperative in the nervous system action. So we're going to have to store some of that, obviously, in like our cerebrospinal fluids and also in our interstitial fluid between our cells, like around the neuromuscular junction. We're going to learn that calcium is also important in blood clotting. So this is very, very, very cool stuff. So calcium is going to be important in stimulating the release of the neurotransmitter from the axon terminal. And it does that by going into the axon terminal once the voltage-gated calcium channels are open. And it's going to stimulate the fusing of the synaptic vesicles to the plasma membrane and thus cause the exocytosis of the neurotransmitter into the synaptic cleft. Voila! So greater frequency of action potentials results in more stimulation of the postsynaptic neuron. So here's some calcium and the synaptic vesicles. So number one, when calcium enters the cell, it binds to a protein called synaptogamin. I didn't say that right. Synaptotagmin. I mixed my G. Synaptotagmin that serves as a calcium sensor and is anchored to the synaptic vesicle membrane. Number two, vesicles containing neurotransmitter are docked at the plasma membrane by 3 snare complex proteins. And number three, the calcium synaptotagmin complex dissipates part of snare and the vesicle fuses, and it, number four, forms a pore to release the neurotransmitter. So here you see, number one, in figure 7.23, the action potential reached the axon terminal, opening the voltage-gated calcium channels. Calcium is going to go and bind to a sensor protein in the cytoplasm and stimulate fusion of exocytosis, or fusion and exocytosis of the neurotransmitter. So once the neurotransmitter is released into the synaptic cleft, it's got to diffuse across the synapse, and it binds to a specific receptor protein. Thus, the neurotransmitter is referred to as the ligand, and this results in the opening of chemically regulated ion channels, which are also called ligand-gated cation channels. All right, that's from a video I show in class. She says it like that, ligand-gated cation channels. They're not always cation channels, though, so stay tuned for that. So we've got these things called graded potentials. So when ligand-gated channels open, the membrane potential changes depending on which ion channel is open. So opening a cation channel results in a graded depolarization called an excitatory postsynaptic potential, or EPSP. However, we can have anion-gated ligand channels that are going to result in a graded hyperpolarization called the inhibitory postsynaptic potential. So if it's the chloride, it's an anion. And then, of course, if we open any type of potassium channel, we know that losing potassium out of our cell membrane is going to hyperpolarize our membrane and make us more negative. So excitatory postsynaptic potentials take us towards threshold. Inhibitory postsynaptic potentials take us away from threshold. All right, so let's talk more about the EPSPs and the IPSPs. So excitatory for E, inhibitory for P, inhibitory for I. So excitatory for E, inhibitory for I, and then the PSPs is postsynaptic potentials. So A, EPSPs, excitatory postsynaptic potentials, move the membrane potential closer to threshold. They may require EPSPs from several neurons to actually produce an action potential. Then we have IPSPs, or inhibitory postsynaptic potentials, which move the membrane potential farther from the threshold and no impulse is produced. IPSPs can counter EPSPs from other neurons. And so the summation of EPSPs and IPSPs at the initial segment of the axon, which is next to the axon hillock, determines whether an action potential occurs. So we can call this summation. And we'll get to that in a second. But here you see 7.24 showing the functional specialization of different regions in a multipolar neuron. Yeah, this is pretty cool. So this is the receptive area up here at the dendrites. You can see that it's getting EPSPs and IPSPs from all kind of different presynaptic neurons. And so they're going to integrate it through that process of summation. So if we add EPSPs together, we can get our threshold reached. If we add IPSPs together, we can go away from our threshold and inhibit the neuron. If we get the same number of EPSPs as IPSPs, nothing happens because they cancel each other out, you know? So if we get an action potential, then it's going to be impulse conduction all the way down the axon towards the boutons, the axon terminals, and the neurotransmitter will be released. Trying to see if it shows any more about the summation, which is really neat. In my A&P book for another like what we call BIOL 168, which is anatomy and physiology, it shows this guy here with like maybe this is going to be IPSP and this is going to be like an EPSP, and it shows like if we get multiple EPSPs, it will cause like small summations towards that threshold. And then, of course, if we get the multiple IPSPs together, it will show small summations away from threshold. So what that's called, if it comes from the same neuron, so like if this neuron right here keeps sending EPSPs, 1, 2, 3, 4, 5, that's called temporal summation, because it's the same neuron that's shooting the postsynaptic neuron many times with an EPSP, and eventually we're going to get an action potential. So that's temporal. Spatial summation is when we have multiple different neurons coming at the cell body. So if we had an EPSP from this guy to the right and an IPSP To this guy from the left, though, that's like spatial because they're from different spaces, different areas on the neuron. And in this case, they might cancel each other out. So anyway, neat stuff. All right, so now we are getting into the neurotransmitters. And I do know that I talked to you about some of those. So let's go to the study guide at this point because there's a lot going on here in this section. And I want to make sure. So we're in chapter 7 now. I'm going to do this for several chapters, like the sensory chapter as well, because there's just so much. So far, we've covered one through 19. So like that graded potential is going to be the EPSPs and the IPSPs. And then we talked about the summation and how the EPSPs and IPSPs can bring you towards or away from threshold. So now we know that once the axon initial segment is stimulated, then we get the transmission of the nerve impulse down the axon. However, if it's inhibited, then, we're quiet. And that's okay, because you want sometimes your neurons to be inhibited. We would be overexcited and absolutely crazy if our neurons were excited all the time. So now we got to look at some different neurotransmitter methods. So we need ligand-gated channels, G-protein coupled channels, and then the second messengers. There's spatial summation. And okay, good. I just got ahead of myself. So you do get to talk about spatial summation and temporal summation, and then some synaptic plasticity. So let's look at the acetylcholine receptors that are going to be nicotinic and muscarinic, and then let's look at those other things afterwards, the ligand-gated channels, G-proteins, and all that. All right, so I'm going to keep that up just in case we need to refer back to it. So I'm going to kind of hop, skip, and jump through these next sections if you didn't gather that already. And again, just really focus on the meat and taters. All right, so acetylcholine is a neurotransmitter that binds to ion channels, the cation channels. And in some cases, the acetylcholine is excitatory, and in other cases, it is inhibitory, depending on the organ involved. So it's excitatory in some areas of the central nervous system, in some autonomic motor neurons, and in all somatic motor neurons. And it is inhibitory in some autonomic motor neurons. And again, it's all right. We need our neurons to be quiet sometimes. We can't be overexcited and overstimulated and everything just going at full force all the time. Okay, so here's the nicotinic and those muscarinic ones. So two types of acetylcholine receptors. We've got nicotinic acetylcholine receptors, can be stimulated by nicotine, and they're found on the motor end plate of skeletal muscle cells, in the autonomic ganglia, and in some parts of the brain. So you'll visit these guys again when you get to the autonomic nervous system, because we learned that pretty much acetylcholine is released by all of the presynaptic neurons in the autonomic nervous system, whether it's parasympathetic or sympathetic. So remember this, okay? And they're nicotinic, as stimulating. Then we have muscarinic. These can be stimulated by muscarine, which is a poison from mushrooms, and they are found in the central nervous system in the plasma membrane of smooth and cardiac muscles and glands innervated by autonomic motor neurons. Now we're talking about the agonists and the antagonists. Let me get to the page here. There's so much. All right, drugs that bind to and thereby activate receptor proteins are called agonists, and drugs that bind to and thereby reduce the activity of receptor proteins are antagonists. So that's what that's talking about there. And let's see. Let me read for a moment, since there's a specific questions about nicotinic versus muscarinic on your study guide. So in your book, under 7.4, on page 182 in this edition, It talks about nicotinic acetylcholine receptors are found in specific regions of the brain. You'll see that in chapter 8. In autonomic ganglia, you'll see that in chapter 9. And in skeletal muscle fibers, which will be in chapter 12. So, boom, boom, boom. The release of acetylcholine from somatic motor neurons and its subsequent binding to nicotinic receptors stimulates skeletal muscle contraction. So that's excitatory in this case. And like I said, in the autonomic ganglia, the presynaptic neuron is going to be excitatory. All right, now with the muscarinic, these are found in the smooth muscle cells, cardiac muscle cells, and the cells of particular glands. We'll see that in chapter 9. And the activation of these muscarinic acetylcholine receptors by acetylcholine released from autonomic axons is required for the regulation of the cardiovascular system. We'll see that in chapter 14. The digestive system, we'll see that in chapter 18, and others. Muscarinic acetylcholine receptors can also be found in the brain. So from what I understand, is like in parasympathetic, where you're resting and digesting, the muscarinic is going to kind of regulate and slow your heart rate down, but it's going to speed up digestion, hence resting and digesting. So our heart rate is slow because we're resting, but our stomach is fast because we're digesting. So that's what I kind of wanted you to get from that. All right, so now we've got to go on. Let me really stick with the study guide here because we could go crazy here. So we're looking at ligand-gated channels, G-protein coupled channels, and second messenger. So with the ligand-gated channels, that's going to be here on page 183. So let's get to that. And that's going to be like the cation-gated channels that I was mentioning earlier in the funny robot voice. And so here, let's take a look at them. Ligand-gated channels. The receptor protein is also an ion channel. Binding of the neurotransmitter directly opens the ion channel. So that's what these ligand-gated channels are doing. So nicotinic acetylcholine receptors are ligand-gated channels with 2 receptor sites for 2 acetylcholines. Binding of the 2 acetylcholine molecules opens the channel and allows both sodium and potassium passage. The sodium is going to flow in, the potassium is going to flow out. Due to the electrochemical gradient, more sodium flows in than potassium flows out. And so this is going to effectively depolarize the membrane. So remember I said that in skeletal muscles, this is going to stimulate skeletal muscles to contract. And in autonomic motor neurons, this is going to be released by the presynaptic neuron, and it's going to stimulate the action potential to occur in the postsynaptic neuron. This is figure 7.26, showing the nicotinic acetylcholine receptors functioning as ion channels. So the nicotinic acetylcholine receptor contains a channel that is closed, A, over here on the right side, until the receptor binds to acetylcholine, over here in number 2. And then what happens is sodium and potassium diffuse simultaneously and in opposite directions through the open ion channel. The effect of the inward diffusion of sodium predominates, resulting in an excitatory postsynaptic potential, an EPSP. Whoop, whoop. So the EPSPs occur in the dendrites and the cell bodies. EPSPs from binding of several acetylcholine molecules can be added together to produce greater depolarization. So that's graded, like that kind of that summation. And this may reach threshold for voltage-gated channels in the axon hillock, leading to action potential. 7.4 table is going to compare action potentials and excitatory postsynaptic potentials. So you can look at that, seeing their amplitude and all that good stuff. And we're going to move on. All right, now we have G-protein coupled channels. You're going to see these a lot. G-protein coupled channels are going to appear. I'm already now on, I'm getting ready to do chapter 13 in my homework prep for y'all. And like literally in every chapter, there's been G-protein coupled channels mentioned. So here we go. So the neurotransmitter receptor is separate from the protein that serves as the ion channel. The neurotransmitter receptor is separate from the protein that serves as the ion channel. Okay? Whereas with the ligand, the hormone bound or the neurotransmitter attached directly to the ion channel. So ligand, the receptor protein, is also an ion channel. Binding of the neurotransmitter directly opens the ion channel. In G protein, the neurotransmitter receptor is separate from the protein that serves as the ion channel. So binding at the receptor opens the ion channels indirectly by using a G protein. So muscarinic acetylcholine receptors interact with ion channels in this way, as well as dopamine and norepinephrine receptors. There are 5 subtypes, M1 through M5, that produce different effects in different organs. G proteins, if you were wondering, it's here in the book on page 185. They are so named because their activity is influenced by guanosine nucleotides, GDP and GTP. This topic was introduced in Chapter 6, Section 6.5. Muscarinic receptors are thus members of a family of receptors called G protein-coupled receptors. Okay, so let me see here. I don't know that I need to go into all the detail on this, because again, there's a lot. So we might want to go ahead and go on to the second messengers, because yeah, whoo, that's a lot of stuff there. And you even get a table that summarizes the steps there, steps of the activation and deactivation. But basically what I was wanting you to get, the meat and potatoes part, is the fact that the ligand receptors, the neurotransmitter directly attaches to it, opening the ion channels, whereas with G proteins, it's going to attach to a receptor other than the ion channel, and a G protein is going to go in, and it's going to open the channel. Let's take a look at this picture. This is a cool picture. This is kind of showing the receptor and the channel, so I like that. That's easy. So muscarinic acetylcholine receptors require the action of G proteins, and here we're seeing it in the heart. So the figure depicts the effects of acetylcholine on the pacemaker cells of the heart. Binding of acetylcholine to its muscarinic receptor causes the beta-gamma subunits to dissociate from the alpha subunit. You can see those here, alpha, beta, gamma. The beta-gamma complex of G proteins then binds to the potassium channel, causing it to open. Outward diffusion of potassium results, slowing the heart rate. See? Easy. Isn't that nice? This is kind of cool. Binding of acetylcholine opens potassium channels in some tissues, which would produce an IPSP, or closes potassium channels in others, which would be an EPSP. So this would be, the resting and digestion. So in the heart, potassium channels are opened by the beta-gamma complex, creating the IPSPs, which is going to cause hyperpolarization and slows the heart rate. Or in number 2, in the smooth muscles of the stomach, the potassium channels are closed by the alpha subunit, producing EPSPs, which are depolarizing, and causes the contraction of these muscles. So in number 1, you're slowing the heart rate by opening the potassium channels, and so you're resting. In number 2, you're speeding up the stomach by closing potassium channels, and so now you are digesting. So yay, muscarinic receptors for parasympathetic nervous system activation. Yep, let's move on. Lots of stuff. Lots of stuff. Okay, so now we've got to get to CAMs, right? These cyclic AMPs, the CAMPs. So let's look at second messengers. Let's find them. Where are you at? Now we're going to talk about monoamines as neurotransmitters. So these are going to be where we'll find the CAMP, the cyclic adenosine monophosphate. So monoamines are regulatory molecules that are derived from amino acids. They include catecholamines, serotonin, and histamine. So catecholamines are derived from tyrosine, and they include dopamine, norepinephrine, and epinephrine. Serotonin is going to be derived from L-tryptophan, and histamine is derived from histidine. So like acetylcholine, monoamines are made in the presynaptic axon, released via exocytosis, they diffuse across the synapse, and they bind to specific receptors. They are quickly taken back into the presynaptic cell, which is called reuptake, and they are degraded by monoamine oxidase. So here's the method of monoamine action and inaction, or inactivation. In step 1, the monoamine is produced and stored in synaptic vesicles. In number 2, just like before, the calcium ion is going to enter as the calcium channels open, and the calcium will then stimulate the fusion of the vesicle to the plasma membrane and the release of the neurotransmitter through exocytosis. The neurotransmitter will then diffuse across the synaptic cleft and it will bind to receptors on the postsynaptic membrane. And then once the signal has been transmitted, the reuptake of the neurotransmitter will occur, and then the neurotransmitter will be inactivated by monoamine oxidase. So none of the receptors for these signals are direct ion channels, kind of like the G proteins. They all use the second messenger system, and this time we're going to see cyclic adenosine monophosphate, or CAMP, which is the most common second messenger for catecholamines. So binding of a catecholamine to its receptor activates a G protein to dissociate and send the alpha subunit to an enzyme called adenylyl cyclase, and it converts ATP to cyclic AMP. Cyclic AMP then activates an enzyme called protein kinase, which phosphorylates other proteins, and then as a result, an ion channel opens. So here in figure 7.31, we see norepinephrine action requiring G proteins. So the binding of norepinephrine to its receptor causes the dissociation of G proteins, binding of the alpha G protein subunit. So this time we're doing the alpha gene, whereas we saw beta gamma in the last example with acetylcholine. So here we got alpha coming in, and it is going to bind to the enzyme adenylyl cyclase, and it's going to activate it to make the cyclic AMP. And cyclic AMP in turn is going to activate protein kinases, which phosphorylates proteins to open ion channels and produce other effects. So that's a lot of work. That's called like signal transduction, and it's amazing that people figure that out. We just have to read about it and learn it, cry a little bit over it, and then other people spent years and years researching and figured this stuff out. It's amazing. All right, so that's how that works. So that takes care of this part of the study guide. So ligand-gated channels, G protein coupled channels, and second messengers. So I just wanted to give you kind of like an overview of how the physiology works of these different neurotransmitters. And I went through and talked to you already about summation. And so your figure where it shows that is figure 7.33, so I'll make sure to show that to you. And then we're going to look at synaptic plasticity. So let's go ahead and get to, you can see how many slides there are. Not yet. We're going to 7.34. Actually, we'll go to 7.33. I was looking at 7.34. All right, so here is spatial and temporal summation. In spatial summation, 2 or more presynaptic neurons release excitatory neurotransmitters to produce EPSPs that can summate in the postsynaptic neuron's dendrites and cell body. In temporal summation, it's a single presynaptic neuron, releasing the neurotransmitter 2 or more times in quick succession to produce EPSPs that can summate. In either case, if the total depolarization exceeds threshold at the initial segment of the axon, it will stimulate the production of an action potential. And so this is just showing an example with EPSPs. Remember, we could get IPSPs coming in as well. And that looks like There we go, postsynaptic inhibition. The lower diagram depicts a recording at the axon hillock of postsynaptic neuron. An inhibitory postsynaptic potential makes the inside of the postsynaptic membrane more negative than resting potential. It hyperpolarizes the membrane. An EPSP, which might have sufficient strength on its own to reach threshold and stimulate action potential, does not reach threshold because it begins at the more negative membrane potential of the IPSP. So again, it's very important to be able to quiet the neurons down. All right, so now let's look at synaptic plasticity and be done with it. I know you're tired of hearing about all this stuff. This is the hardest chapter in this unit, so that's why I recorded this one first. So repeated use of a neuronal pathway may strengthen or reduce synaptic transmission in that pathway. The ability of the synapses to change is called synaptic plasticity. And this is an important concept because this is going to be how like a lot of learning is going to occur with plasticity and habit formation and breaking habits. So when repeated stimulation enhances excitability, it is called long-term potentiation. And this is found in the hippocampus of the brain where memories are stored. And it's associated with the insertion of AMPA glutamate receptors. It improves the efficacy of synaptic transmission that favors transmission along frequently used pathways. Long-term depression occurs when AMPA receptors are removed, and this suppresses the further release of the neurotransmitter, and thus learning is impaired. But this also can occur, like I said, when you're like breaking bad habits. So give yourself a little bit of time when you're breaking bad habits. Because you have to literally break neuronal connections in order to do that. And then also with creating good new habits, you have to make neuronal connections. And so, give yourself some time and patience as you do that. But just know that this is how it occurs. It's called plasticity. So both long-term potentiation and long-term depression depend on a rise in calcium ion concentration within the postsynaptic neuron. A rapid rise of calcium ions leads to long-term potentiation, and a smaller but prolonged rise leads to long-term depression. So synaptic plasticity also involves the enlargement or shrinkage of dendritic spines. All right, and then it goes into the postsynaptic inhibition, which makes sense because that can lead to that long-term depression. And so I was thinking about GABA as I was talking about it earlier, and that is mentioned here in the book. So the neurotransmitters glycine and GABA hyperpolarize the postsynaptic membrane, and they make the inside of the membrane more negative. And that's how GABA helps to calm you down. And there's your synaptic inhibition. Sometimes a neuron synapses on the axon of a second neuron, inhibiting the release of the excitatory neurotransmitter, and that is because it inactivates calcium ion channels. And so we see this in the action of opioids and pain reduction. Also, like if you hear about medicines such as Xanax or alprazolam, they increase GABA and can cause long-term depression in the neurons. And we're done. So thank you all very much for putting up with me during this long chapter here, and good luck with your studying, and I appreciate you.